Recombinant DNA technology. In order to study a gene, we introduce our gene of interest from an organism into a vector. We then introduce this recombinant DNA into the host cells, allowing it to multiply. This DNA can then be isolated, purified, and studied further for better understanding of its functions. This technology is called recombinant DNA technology. The E.coli system. The bacterium Escherichia coli, or E. coli, is an extremely convenient tool for molecular biologists to study the effect of genetic manipulations. We can observe rapid results which can be quantified easily, since it is a single-celled organism. It has a simple genomic makeup. It can reproduce and grow very rapidly. In order to understand what protein a gene codes for and study that protein, scientists cloned the gene into a study organism. The E. coli cell is an excellent tool through which we can understand the genes responsible for certain traits. Gene of interest. This is the desired gene from a study organism. This gene is selectively removed from the genomic or plasmid DNA and can be cloned into a host organism for further analysis. Vector. We use a carrier known as a vector to transfer a gene from one organism to another. These vectors are engineered to contain the following parts: an origin of replication to facilitate the expression of the gene, multiple cloning sites which contain numerous sequences recognized by restriction enzymes, antibiotic resistance marker that allow the organism to thrive in a medium containing specific antibiotics. The most common vector is a plasmid. Plasmids are the circular double-stranded DNA of bacterial cells. A popular plasmid is PBr322 of E. coli. Other popular vectors include bacteriophage. Phage vectors can carry up to 20 kilobases of foreign DNA as an insert. Cosmid. These vectors can carry inserts of 30 to 50 kilobases in size. Bacterial artificial chromosome. BACs can carry anywhere from 50 kilobases to 250 kilobases DNA inserts. Yeast artificial chromosome. YACs can carry 500 kilobases to 3000 kilobases DNA inserts. Restriction enzymes. Restriction endonucleases are enzymes that act as molecular scissors to allow the transfer of a gene from one strand of DNA to a vector. They recognize specific sequences of DNA around 4 to 7 nucleotides long with a palindrome arrangement, meaning that it reads the same in both strands. For example, EcoR1 recognizes GAATTC in the 5' to 3' strand and CTTAAG on the 3' to 5' strand. It catalyzes the hydrolysis of nucleotides on the DNA backbone, essentially cleaving the DNA from within the molecule. This leaves a few unpaired nucleotides on one end that is free to hybridize to another strand. Such ends are called sticky ends. The enzyme ECOR1 generates sticky ends, leaving a 3' overhang. Some other enzymes, such as HPA1, do not create any overhangs. These ends are called blunt ends. A vector has multiple cloning sites with multiple restriction sites for different enzymes. The DNA can be digested with restriction enzymes and mixed together. The complementary strands then hybridize. Nomenclature of restriction enzymes. Restriction enzymes are named after the bacterium from which they are isolated. ECOR1 is isolated from Escherichia coli. The first letter stands for the genus name of the organism, that is Escherichia. The second and third letters represent the species name of the organism, that is coli. The fourth letter stands for the strain designation RY13, and the fifth denotes the order in which the enzyme was discovered, meaning ECOR1 was the first enzyme isolated from the bacterium. Antibiotic resistance marker. In order to check if transformation has occurred, we use marker genes. A dominant selective marker, such as an antibiotic resistance gene, is constructed into the vector. If a host cell takes up the vector, it takes up the gene of interest in addition to a gene that provides antibiotic resistance. Hence, it is able to thrive in media containing that particular antibiotic. This allows us to select the cells that have taken up our gene of interest. Gene transfer. Once the gene has been engineered, it can be introduced into a host cell via one of the following methods. The three most popular modes of gene transfer are transformation, transduction, transfection. Transformation. A bacterial cell is made to absorb the genetic material through the cell membrane and expresses the newly received DNA. Transduction. A virus can be used to transfer the genetic material to a bacterial cell. Bacteriophages are viruses that infect bacteria. The virus carries the gene of interest and injects it into a bacterial cell. This DNA then integrates with the genetic material of the bacteria and replicates it as its own. Transfection. We can also deliberately introduce genetic material into eukaryotic cells through non-viral methods. When an electric field is applied, the permeability of the cell membrane increases. This allows the DNA molecules to easily enter the cells. This mode of transfection is known as electroporation. Polymerase chain reaction. When some DNA is isolated and purified from the source, we require larger quantities to conduct studies. So, we can use an amplification technique known as the polymerase chain reaction or PCR to generate large amounts of a selected DNA sequence. A fragment of DNA, about 100 or more base pairs in length, acts as the template. We then add primers of length 17 to 30 nucleotides. The forward primers are complementary to the beginning of the template and the reverse primers are complementary to the end. The deoxyribonucleotides DATP, DCTP, DGTP and DTTP make up the nucleotide pool. A polymerase enzyme is employed to extend the strands. The first and most commonly used one is Taq polymerase from the organism Thermus aquaticus. It is heat resistant and can withstand high temperatures. There are 3 steps in each PCR cycle: denaturation, annealing and extension. Denaturation. When the DNA is heated to about 94 degrees Celsius, the double-stranded DNA is separated into 2 strands. Annealing. This process occurs at 55 to 65 degrees Celsius. The forward and reverse primers bind to the 2 separated strands. Extension. At around 72 degrees Celsius, the polymerase adds nucleotides complementary to the strands in the 5' to 3' direction. This way, an exponential amplification of the DNA is observed with each cycle. An optimum PCR reaction is around 30 cycles. Some applications of PCR include diagnosis of genetic diseases, DNA profiling, rapid detection and diagnosis of infectious diseases and pathogens that are difficult to culture, research laboratories, gene manipulation and expression studies, forensic medicine, and gene cloning. Production of human insulin. Using recombinant DNA technology, we can engineer genomes to produce specific gene products. This has helped us manufacture a wide range of products on a large scale. A good example of the protein produced through this technology is synthetic human insulin. The genes coding for human insulin A and human insulin B are inserted into the beta-galactosidase gene in the plasmid DNA along with a tetracycline resistance gene. The plasmid is inserted into bacteria through transformation. The antibiotic tetracycline is added to eliminate the untransformed cells. Only cells that take up the recombinant DNA can thrive in a medium containing tetracycline. The proteins are secreted with beta-galactosidase as one chain. The beta-gal part is cleaved off. The A and B chains are joined together to form the full-fledged human insulin. Through recombinant DNA technology, proteins for research, diagnosis and treatment can be manufactured. Some medically useful recombinant products like human growth hormone, calcitonin and vaccine can be produced by recombinant DNA technology.